Hello, everyone. Welcome to MACSMIN 2025. It's a great pleasure to introduce Professor Erika Flapan, our collaborator, who will talk about topological complexity in protein structures. Over to you, Erika.
Thank you for the invitation, and I look forward to hearing Pavel's talk after mine. I'm not driving in Kyrgyzstan, so there we go. Anyway, I'm going to talk about topological complexity in protein structures. So let's start in the beginning. Whoops, my controls don't work. There we go. Okay, until the 1990s, all native protein structures were assumed to be topologically simple. That is, even though it maybe looks complex on the left, if you sort of smooth it out a little bit, you see that it's just a line with wiggles in it. And in fact, in 1994, the biochemist Mark Mansfield postulated that knotted proteins couldn't exist, that this is basically what proteins would be, unlike DNA, which was much longer and was known to be knotted, possibly knotted. However, in 1994, Liang and Mislow, who is a chemist, not a biochemist, identified the first knotted protein by including disulfide bonds and copper atoms as well as the backbone. So in this picture that I have here on the left, the backbone is made up of parts that are yellow and parts that are red. The reason I've colored some parts yellow is because I then erased them in the picture on the right. And you can see that there are these copper bonds labeled with these black dots, as well as some disulfide bonds. And so now if you look at this whole structure, it looks complicated, but now I'm going to erase the yellow parts and the turquoise parts. And what it leaves me with is the picture on the right. And if you trace it around, starting, say, here, you go over and then under and then over and then under and over and under, and you're back to the start. This is a knot. So this showed that knots could exist, but it wasn't until 1996 when Takusagawa and Kamatori found the first protein with an actually knotted backbone. That is, they didn't rely on any bonds, like in this example, in order to have the knot. And currently, there are over 200,000 proteins with experimentally solved 3D structures on the PDB, and of them, 1,612 have knotted backbones, and that's as of the last time I checked. There may be more now. So obviously, proteins can exist with knotted backbones, and they're not a rarity, even if they're not a high percentage of proteins. So furthermore, using machine learning techniques, the recent databases of AlphaFold and EMSFolded predicted the three-dimensional structures for hundreds of millions of proteins, which includes 700,000 proteins whose backbones contain complex knots that have not been previously found. So these 700,000 knotted proteins don't necessarily exist. These are being predicted by machine learning based on a learning sample of the existing ones. But in any case, it gives us the idea that there could be lots and lots of knotted proteins. So here are some of the ones that exist so far. And you can see I'm labeling them according to the way knot theorists label knots. So if you're not familiar with this, let me explain. So knot theorists look at a knot and figure out the fewest number of crossings that it can be drawn with. A projection is a drawing of it. And then there are tables that list all the knots with up to that many crossings. So for 3 crossings, there is only 1. You have this knot and its mirror image, which are called trefoil knots. For 4 crossings, there's also only 1. This knot, its mirror image is the same as it up to a deformation. For 5 crossings, there are actually 2 different 5 crossing knots, only one of which has been found in a protein. and it's this one, and it also has a mirror image, but that hasn't yet been found in a protein. For 6 crossings, there are 3 knots, and 2 of them have been found. This 6,3 is the same as its mirror image, so we don't list a plus or minus, but 6,1 has a positive and negative form. For 7 crossings, this is the only one that's been found. For 8 crossings, this is the only one that's been found. Now, this, of course, has 6 crossings, but it's not considered prime, which means that it can be split into 2 knots. So you can see it's split into this knot with 3 crossings and then another knot with 3 crossings. So we write it as 3,1 plus 3,1, right, rather than expressing it with the number 6. Anyway, you can see there's actually quite a lot of knots that have been identified in proteins so far. Now, these knots, as you see in the pictures, are actually not circular because most proteins are not circular. However, the energy required to unthread them is large. So it's as if the knot is stuck in there, but from a topological point of view, we could just unthread them. The ends are not fixed, so we can unthread them. So modeling them as a knotted chain, the way they are as proteins, is not a good option from a topological point of view because topologically that would make them trivial. So to model them, we actually want to join the ends to create a closed loop. And there are various techniques to do this, and you want to do it in such a way that it really captures the knot that exists in the backbone. And so the way that is most commonly done is that you extend the ends I write to infinity. That's what people say. It's actually, of course, infinity doesn't exist, but it's very far. You extend the ends very far and join them at the very far distance. And you do this in hundreds of directions. And then you choose the knot type that occurs most frequently. And you don't have to understand what this means, but they use polynomials to identify which knot type it is. And for example, when I say you extend them in hundreds of directions in order to choose the type that occurs most frequently, on the bottom here, I'm showing what you don't do. So here's a protein. Here's a simplified view of it. You don't join these ends by threading in and out and in and out, because that wouldn't be the one that occurred most frequently. So the one that's going to occur most frequently is the knot that's going to occur most frequently is what you would get if you extended them out away from the core of the knot. And you can see that you can do that in many directions for this example, and you'll get this same knot. It wouldn't be this one. So, all right, let's see if we can, there we go. Okay, so protein knots aren't the only type of entanglement. For example, there can be protein links. And there are a lot of protein links, but most involve what are called cofactors, which are additional compounds. As you see here, the backbone is green. And here I have this iron and this cofactor here, which is attached to the backbone. And so we get a link, and the link here, you can see it as a green loop and a purple loop that are linked. And we call this a Hopf link, which they're linked in the simplest possible link. And most protein links involve just a simple link like this. Now, even though the most common type of protein links involve cofactors like this, it is possible to have a link without a cofactor, with just the protein backbone, if we include disulfide bonds or metal ions in order to create loops. So here in the bottom, the black I've drawn is the protein, a simplified picture of a protein, and the yellow are the disulfide bonds or metal ions. And so because of those ions or bonds, you can see we form a loop here, and we form another loop here. So even though we're only involving one protein, no cofactors, we actually can have a link. And so you would see this link if you wanted to by removing this piece of the backbone. So you have actually, again, a Hopf link. That is that simple link. So in 1995, Liang and Mislow identified this protein link with cofactors. So here we have many cofactors, and here I've drawn the backbone in a mixture of red and green with the intention of erasing the red so that we just have the green on the right. And then I have these blue cofactors which form rings. And so this is called a keyring link because the green is like your key ring and the blue are like the rings of are the keys on your key ring. So this is a more complicated link than just the simplest type. And in fact, we can have an even more complicated link. This is a spherical protein link. It's a chainmail protein capsid, which protects the bacteriophage HK97. And it's made out of 12 pentagonal rings. You can see I highlighted one of the pentagonal rings in blue here, and 60 hexagonal rings. You can see most of these are hexagonal rings. And they're all linked together in a kind of chain mail. And the stability of the capsid comes from the linking. And this kind of stable capsid has the potential to be used for drug delivery. If you imagine you have this capsule, if you put a medication inside of it and you want to deliver it to a specific part of the body, like where you have a tumor, you don't want it to become systemic through the whole body. You just want it to go, say, to the colon. Then you have to sort of program it so that the link comes apart, so that some of the rings are cut and it opens up and the drug goes when it just gets to the part of the body that you want it to go to. So such types of links have a lot of potential. In fact, knots have a lot of potential as well, because knots, they have found, are stretchy. If you imagine like a knitted sweater, which is stretchy, it's stretchy because of all the little knots, which are what the knitting is made up of. And so there's work being done in material science to design, for example, plastic bags, like for supermarkets or whatever, that are made out of a knotted, sort of a knit kind of material that would be more stretchy and less likely to break. So knots and links have a lot of potential applications. But in any case, even these are not the only types of entanglement that we can have. For example, if we include disulfide bonds and metal ions, we have various other types of entanglement. And so here I have a collection, which I'm going to go into more detail about each of these as follows. So this is called a slip knot, and a slip knot is an unknotted chain. So if you imagine pulling the blue end, you can untie this slip knot, kind of like when you tie your shoes. It's not really a knot, it's a bow, but it's a bow that when you pull becomes undone. But if we were to cut this blue arc, as I've done in this picture here, we end up with a knot. So that's the definition of a slip knot. A slip knot is an unknotted chain where if I remove a piece of it, then it becomes knotted. And here we have some other types of entanglement that I'm going to talk about some more in what follows. So a theta curve is a graph that looks like the Greek letter theta. That's why it's called a theta curve. And let's just go back for a second. Here you can see that it looks like the Greek letter theta, but it can be entangled. So Selkowska et al. Found these theta curves, which you don't have to worry about the way that she's labeled them. She looked at the protein bond, the protein backbone, joined the two termini in the way that we join the termini when we're defining protein knots, and then included a disulfide bond. And so when you do that, you end up, again, let's just go back to see a simple picture. Here, you can imagine that this black arc is the protein backbone. Here, the ends are joined at infinity in the probabilistic way that I described. And this is this is a disulfide bond that joins them. We could also have both of these be disulfide bonds or metal ions, but that's not what Solkowska did. So then one more thing back here. So if you look at this, you can think of this as having three pieces, say a red arc here. This could be a blue arc going on the top, and then a green arc like that. And so she drew these so that there is a red arc, as you see, a blue arc, and a green arc. And so you can see that these are not just a simple theta that looks like a Greek theta. In fact, in this one, you have the red and blue together make a knot, and you have this green arc. This one's a little different. The red and blue together again make a knot, but the green arc is parallel to the blue arc. The red and blue make a more complicated knot, and here you have the green. And similarly here, the red and blue, or for that matter, the green and blue, either one makes a knot, and then the other one threads through it. And then here are some more that are similar. Well, these ones, the red and blue, again, makes a knot. And here, this one is a much more complicated knot. So this might lead you to think that the only way you could have a theta curve, which is not just a planar one that looks like the actual Greek letter theta, is by having two of the arcs, say the red and the blue, forming a knot. But in fact, that's not true. This is an example of an entangled theta curve, meaning can't be deformed to lie in the plane to look like a theta, but it doesn't contain a knot. So if you look at this and you imagine erasing the green, you'll have the red and the blue, and it won't be unknotted because the red and the blue can unthread. And similarly, if you just look at the red with the green, it's just a little twist. And if you just look at the blue with the green, it's just a little twist. So this doesn't have any knot. If you look at any two of these arcs, it isn't a knot. And yet it cannot be deformed to look like a planar curve. It would be really interesting if such a theta curve could be found in a protein, but so far that hasn't been the case. But I'm guessing it will be found if we continue looking, because proteins are increasingly complicated as we consider bonds, the disulfide bonds, as well as metal ions. So continuing on looking at the different types of entangled structures that have been identified, there are cyclotides, which are small protein loops, so they are actual loops. Here, the black is the protein backbone, and the red are the disulfide bonds. And it may look like it could be deformed in the plane, but what I actually am showing in these pictures is that it can be deformed to look like a Mobius strip. So as I go from this first picture to the second picture, what I'm going to do is I'm going to slide this red bond going from the 3 to the 6 to the left, while I slide the one from the 1 to the 4, sliding the 1 along here and the 4 along here to the right. Of course, I'm not saying that you're sliding these as actual disulfide bonds. Rather, I'm looking at the topology of this graph by imagining that it's incredibly stretchy and that I can deform whatever I want. And then what I did is I took the black and made it into a kind of figure 8 so here's the crossing of the figure 8, where the 3 and 6 bond, 3 to 6 is over here, and the 2 to 5 is on the left also. The 1 and 4 is on the right. And then I'm going to kind of fold the two big loops of that figure 8 down together in the back of the screen so that the 2 and 5 is here. And you see it really does look like a Mobius strip. And this is called a Mobius ladder. And this cannot be deformed into the plane. In fact, this graph is what's called a K33 graph, the bipartite graph with two groups of three vertices. So it's not quite so obvious to see, but you imagine the 1, 3, and 5 as one group of three vertices, and the 2, 4, and 6 as another. And if you know about the Kuratowski graphs, you'll know that the K33 graph, this graph cannot be deformed to lie in a plane. So that's the cyclotide. The what's called a cysteine motif, or more commonly a cysteine knot motif. I don't like calling it a cysteine knot motif because it doesn't have a knot in it, but that is often what it's called. Also has the form of a Mobius ladder, but in this case it's open. It's not a closed loop. So this is the way it's normally pictured with these bonds. And now again, we can slide the 2, 5 down. So it's at the bottom there instead of being here. And then I can sort of shorten the 1, 4 so that it pulls this part of the backbone over to the left and causes this twist there. And then I can sort of flatten out this loop here to be this small loop, and I have this, make this into a big loop, and then you can pull up slide the 2 over to here to pull it up over here to get the 2, 5 bond, and here you have your 3, 6, and you can see that it again looks like a Mobius strip, or in this case a Mobius ladder, but here we have the N and the C need to be joined in order to complete the ladder. So these two are interesting because of the fact that they have the form of a Mobius ladder. A lasso is an interesting example. In a certain sense, it's geometrically interesting rather than topologically interesting. So in this case, you create a loop with a disulfide bond or a metal ion. So here you have the loop. This creates a loop, and then it has tails. Here's one, in this case, short tail and a longer tail. And at least one of the tails is going to be piercing through the loop. So topologically, this seems trivial because I could just unwind it. But in fact, the tail has side chains that create bulkiness, which prevent it from sliding through. It's similar to what people in chemistry know as a rotexan, where you have a kind of barbell structure with bulky ends and a loop in the middle that can't go off because of the bulky ends. So in this case, this tail can't become unthreaded through the loop because of the bulkiness there. Okay, so there are lots of different lassos. And in 2021, Solkowska made this table of lassos with a particular way of referring to them. We don't need to worry about this particular terminology. Here, the blue ones have all been found, and the gray ones are conjectured to exist because symmetrically they seem like they should. What you can see, if we look first in the center, this wouldn't be considered a lasso because neither end is threaded through the center. But here you can have threading in various different ways where we're labeling whether it's the C end or the C end or the N end, and whether it's passing through from what would be considered the positive side of the loop or from the back, the underside, and that's what that's referring to. And what you can see is that all of these lassos sort of have two possible things they do. They either wind with one or both ends winding around the loop, possibly many times, like in this example, you have it winding many times around. And then that you also have the possibility that it sort of goes through what we might consider trivially, like just a little end pokes through, as in this case, pokes through and then comes back. But again, it's not, even though topologically that would be considered trivial, it can't just undo because of the bulkiness of the backbone. So all of these examples are made up of either twisting one tail around the loop or going back and forth through the loop, or some combination. Like, for example, here you have twisting and going back and forth through. But in all of these examples, topologically, if you ignore that, the fact that it's geometrically stuck there, topologically, they would be trivial. So they're still considered entangled because they're stuck there, but it's kind of noteworthy that it's not the same as the examples I was giving of knots and links and theta curves and even the cyclotides and so on. More recently, Salkowska found some entangled lassos by including disulfide bonds. So here too that her group found. So here you can imagine if you were to join the ends of, so the backbone includes these red ends. If you were to join the ends, we would have a knot here, but actually what we're going to add this disulfide bond. And if you add the disulfide bond to create the loop of the lasso, then the loop consists of the black together with the orange. And you can see that the black together with the orange isn't actually knotted. In order to obtain a knot, you actually need to join the two ends together. And here on the right, you have a lasso, which has a loop, which is again the black and the orange, where this end goes back and forth, as we saw in the previous cases. But this one is interesting because the tail itself has a knot in it. Again, that knot, in order to be held there topologically, we would need to join the ends. So these are both very interesting examples of entangled lassos. They're not like the ones that we saw in this previous slide from 2021, because none of these had any topological entanglement. However, they don't involve a knotted loop of the lasso, and the knot itself is only there if we join the ends. So very recently, Anasova, Curlin, O'Donnell, and I have been looking at spatial graph proteins. That is to say, we're looking at the backbone together with all of the metal ions and disulfide bonds and not joining the terminals, because the terminals are not actually joined as part of the protein. It's only a way, it's like a topological model that we do in order to include knots. We'll join the ends. But if we're going to consider the protein just in and of itself as what we call a spatial graph, that is to say, a graph living in three-dimensional space as just an object itself with ends that stay there, then we're not going to join them. But what we are going to do is we're going to include the metal ions or disulfide bonds, which are in fact part of the protein. So I have two examples here. Here's one example that we looked at. And what you can see, if you sort of erase some of the bonds and include others and color part blue, part red, and part green, we get a theta curve. And here I simplified the theta curve. And you can see that the theta curve includes a knot where remember, a theta curve is made up of three arcs: a red arc, a blue arc, and a green arc. And here, if you look at the blue arc, you need to include this metal ion chain here to get the rest of the blue. So I have a blue arc that goes around like this, a red arc that goes around like this, and then the green arc was going through the metal ion over here. And we have a theta curve which is knotted, just as we saw some of the previous examples of knotted theta curves. So it's not one of these ones that's entangled but not knotted that I hope to find but have not found thus far. But it's another example of a knotted theta curve. But also in this same protein, we can look at it with a different focus and find two different interesting entangled lassos. So here what I did is I just included this one metal ion. And if you look at the black curve, so the black curve is going to be the loop. If you include this, this orange joining with a metal ion, you see it's a loop. The black curve is not knotted, just as the lassos that Sulkowska had are not knotted. But this is an interesting lasso. I'm simplifying it here in the next picture. So the red tails are included here. So I've simplified it. You see that the black is not knotted. It forms what I would call a fish form, which this also is kind of a fish form, but where the tail of the fish is flipped up. Anyway, if we were to join the ends, it would be knotted, but I'm not calling this a knotted lasso, because as a lasso with the ends not joined, it does not contain a knot. But it is interesting in the fact that it has this sort of fish-like picture with one tail cutting through it, which would be knotted if you were to join the ends. So it's entangled. This is a different lasso that's contained in this same protein. So now I'm choosing a different one of the metal ions that I want to focus on. And again, the loop of the lasso is black together with this orange edge here. And we see that the black is not knotted. And then we have our tails, which are threaded through. They're interesting because they sort of link together, the two tails. So here is a simplified picture of this lasso where you can see the black and orange is the loop. It's certainly not knotted. And neither one of the tails is knotted, as in the Sulkowska example, but the tails link through, well, one tail links through the black loop. The other tail does not link through the black loop, but sort of twists through the first tail. So again, if we were to join the tails at infinity, we would have a knot, as you would in the Sulkowska example, but we don't want to do that. We want to look at it just as the spatial graph that is the protein itself. So these are entangled lassos that are not actually knotted, but there is a knotted theta curve, and these are all contained in this one protein. Now, here's another example. It looks quite a bit more complicated. But here I simplified it. So again, only including the part that I wanted. So I erased here. I didn't erase very much. I erased this piece of the bond joining this metal ion. And here I colored what's going to be the loop black and the tails red. And so you can see that the black loop, if you start here, you go around here, around. I think I didn't follow with the arrow very carefully. And then I join the two ends here with this, with these metal ions. And this red tail is a piece of a tail together with a metal ion and then the other piece of the tail. Here I'm simplifying it to make it easier to see. So I sort of took out the kinks and smoothed it a little bit. And here I simplified it more still. And simplified this way, what you can see is that we actually have a knotted loop here. So if I look at this the black arc going around together with the orange, we get actually a knotted loop. And then the tail, the two tails, this one links through this piece of the loop, and this one twists around the other piece. And so this may be the first example of a lasso that actually has a knotted loop where we're not joining the ends at infinity. It's the lasso with the knotted loop is actually living inside of the protein as it exists. So these are examples of spatial graph proteins which have actually interesting subgraphs that hadn't been found before. This one in particular with the lasso with a knotted loop. All right. So what I want to do now is I want to go back to, well, actually, before I go back, one more example. These are metalloproteins where you have lots of metals that are joined to your protein. And this is interesting. Here's the example. It looks like a cube inside of a circle. And here my colors are just to help us see the deformation. So I simplified it. I simplified it going from the first to the second. And then I took this green part and made it into a sideways figure 8. You can see it crosses over here. And I erased the purple arcs so that you can focus on the green figure 8 together with the orange arcs. And then going from here to this step, I sort of pushed the two ears of the figure 8 together behind the screen and saw that we're going to have the 1, 5 down there, the 2, 6 is a little further up, the 3, 7 here, and the 4, 8 is going to now be at the top. And if I turn it over, I end up with this picture of, again, a Mobius ladder, but it's different than the cyclotides because we actually have 4 rungs on our ladder, but it still looks like a Mobius strip. So what's interesting about Mobius ladders is we can think of the graph of a Mobius ladder as an abstract graph in space, and you can see that the green is an unknotted loop, but we can think about it abstractly just as a loop in space. And what we can see is that if you go around that loop, you'll pass through 1, 2, sorry, I am going, well, I'm numbered this like this. So let's look at it like this instead. Here, I'm going to go, if I pass around the loop, I'm going to go 1, 2, 3, 4, 5, 6. So what you see is that the rungs of the ladder are joining opposite points on the circle. So if you go from 1 to 4, you go through all of the rungs once before you come back to the first rung to the opposite endpoint. So it's like this. So as an abstract graph with 3 rungs, we're going to call it M3. It looks like I could think of it as a hexagon, or I could think of it as a circle. And so if I embed this in space in the way that I have here so that it looks like a Mobius ladder, we call that Mobius form. So this here is the abstract graph where there's no vertex in the center. I just don't know how I'm having my edges embedded. We could embed the edges in a different way. Here's an example where this one goes in and out. We could imagine that the outside ring is knotted. That would be yet another way. So any one of these Mobius ladders with any number of rungs can be embedded in space in infinitely many different ways. You can knot up the outside circle, or you can twist together some of the inside rungs, and we would say they were not of Mobius form. But if it's embedded in space in such a way that it looks like a Mobius ladder like these, then we say it has Mobius form. So far, we don't know any proteins or metalloproteins that contain an MN graph, even with just 3 rungs, which is not in Mobius form. And you might expect that just like not all protein knots have the same knot, you might expect that not all protein Mobius ladders have Mobius form, but we don't know any examples of that. So that, like the theta curve, which has no knots but isn't planar, is something we would seek to hopefully find at some point, but so far we haven't found anything like that. So these are all interesting types of entanglement that we might hope to find. So now I want to go back to talk about knots in a little more detail. So it's not known whether protein knots actually serve a purpose or if they're just a random occurrence that's been preserved by evolution. So if they were a random occurrence, we wouldn't expect the same protein to be knotted in distinct organisms, because we might think that, as the organisms have evolved over over thousands of years, some would evolve with a knot, some would evolve without the knot, and so on. And yet this is not the case because, for example, carbonic anhydrase containing the 3,1 knot has been found in humans, bacteria, and algae. And class 2 ketyl acid reductoisomerase containing the 4,1 knot has been found in E. coli and in spinach. And ubiquitin hydrolase containing the 5,2 knot is found in humans and in yeast. So all of this points to the possibility that knots are not a random occurrence, but rather serve a purpose, even if we don't know exactly what the purpose is. So protein knots could actually turn out to be important for medicine, both for good and bad reasons. So in Parkinson's disease, it seems to be caused by the degradation of ubiquitin hydrolase. As it passes through a narrow pore, it becomes unfolded and degrades. And the 5,2 knot, which is normally in ubiquitin hydrolase, would make it difficult for it to thread through the pore. So this is not what it looks like. This is a schematic for you to imagine a thread going through a needle, and if the thread has a knot in it, it can't pass through the eye of the needle. So we could imagine that the 5,2 knot is actually serving the purpose of preventing ubiquitin hydrolase from passing through this pore and degrading. So we don't know if that's what, if not having the 5,2 knot is what causes Parkinson's disease, but it's one hypothesis of what could be happening. So here, the idea is if we could understand better how and why the 5,2 knot occurs in ubiquitin hydrolase, we might understand how to cure Parkinson's disease or how to prevent it. On the other hand, more often, protein entanglement has been known to be linked to misfolding diseases, including Alzheimer's, ocular pharyngeal muscular dystrophy, and certain types of cancers, including a certain type of breast cancer. Furthermore, some of the spout family of proteins, which all contain a deep trefoil knot, are known to cause genetic diseases. So here, again, if we could understand the knots, how and why they occur in these particular proteins, we might be able to prevent or cure some of these diseases. So we might wonder how these knots fold. So William Taylor in 2007 developed what was called the twisted hairpin theory of how proteins knot. And what his theory said is that first a knot twists and then one of the termini threads through the loop. So here you can see, if you imagine a single twist and then the C terminus threads through the eye of that, you imagine we can call this a twisted hairpin. It threads through the eye of the twisted hairpin. And if you join the ends with the C in the end, you get a 3,1 knot. If the twisted hairpin had 2 twists before the C threaded through it, we would get a 4,1 knot. If it had 3 twists, we would get a 5,2 knot. And if we had 4 twists, we would get a 6,2 knot. So a 6,1 knot, excuse me. So at the time, 2007, when William Taylor developed this theory, all protein knots that were known were either a 3,1, a 4,1, a 5,2, or a 6,1, which are all what's called twist knots. Twist knots all have the form of this series of twists together with a kind of hook. So it's easiest to see in the 5,2, you have a series of twists, you could have more twists, and then you have a hook. So these are all called twist knots. Right now, there are more knots that are known that are not twist knots, but this was a reasonable theory for how all knots formed in 2007 when William Taylor formed this theory. However, Bollinger, Sulkowska, and al. did molecular simulations of the 6,1 knot, which is a twist knot. But what they found doing these simulations of how the DEH protein might fold was that it did not follow the twisted hairpin pathway, even though that was a conceivable pathway. And so instead, what they found is in their simulation is that first, a green loop, I mean, the loop isn't green on the protein, but in the picture it's green. A green loop forms, and then a red loop forms, and the blue end is like hooked. And in most threadings of proteins, it begins with a hooked end, which then opens up. So the two loops form and they align. And then they found two different paths. So this step 3A followed by step 4 has the path that the red loop adds one more twist. Here's only one twist. It has one more twist. And then the green loop flips over the red loop. So here was the green loop. It flipped over the red loop. And then the blue end threaded through both loops at the same time and opened up to form the 6-1 knot. The other path that they found was that after step 2, the red loop twisted up once more by itself without the green loop flipping over it. And then the blue end threaded through the red loop, followed by the green loop flipping over both the red loop and the blue end. And then the blue end opened up to form the knot we found here. So according to these simulations, this was how the 6-1 knot formed rather than through the twisted hairpin theory. So then together with Adam He and Helen Wang, we generalized the steps of this simulation to see all the different possible knots that could form in this way. And the way we did this, we had the two different routes. And what we did is we said, well, a red loop and a green loop form with up to 2 twists. So if we go backwards for a second, we see that in this case, where they were looking at the 6-1 knot in DEHI, they had the red loop had 2 twists, the green loop has 1 twist. So in our case, we're allowing the red loop and the green loop each to have up to 2 twists. And you can see that they could twist one way or they could twist the opposite way. And so we allow either of those. Then the blue end passes over the end of the red arc as it approaches the loops which are aligned. And then there are 2 possibilities. Either the green loop flips over the red loop and threads the blue end, and then the loops align and the blue threads through the red loop, producing my final knot. Or the blue end threads through the red loop first, and then the green loop flips over both the red loop and the blue end, producing my knot. So what we wanted to know is if we allow up to 2 twists, and this blue end could go either in front of or behind the red end, we wanted to know all the possible knots that could happen in this way. So we developed a notation here where we indicated we listed the red loop first and indicated either an R if the red loop is in front of the blue and on the right side of the red loop. So the R means right side. It doesn't mean red. It means the right side of the loop goes in front of the blue. And then we listed the green one and indicated if the right or the left side of the green one was in front of the blue, that's here. Then we indicated a plus depending on whether the slope of this black crossing was plus as it went over or negative if the red one was the one that went over, then the slope of the black one would be, the slope of the overcrossing would be, the red one would be a minus. And then the numbers indicate the number of twists where, again, it could be a plus or a minus depending on the slope. So we refer to all the possible configurations we could get in this way as configurations, all the possible results. And so what we did is we drew them in a more stylized way by separating out, by pulling off the green away from the red. Topologically, it would be the same if we didn't allow it to undo. And so we see we have 8 possible images here, where the A and the B can be up to 2 twists, either positive or negative twists. The plus is if the black goes over the red. The minus is if the red goes over the black. And then we indicate if here, for example, it's the left side of both the red and the green that are in front. Here it's the right side of both the red and the green that are in the front. And here you have the left one of the red, the right of the green, and you always list the red first. So we get 8 different possibilities here. And then we figured out all the possible knots you can get where we allowed up to 2 twists in each loop. So we're only allowing up to 2 twists because of energetic restrictions, that the more twists you have, the more energy required. So we can get a lot more knots if we allow more twists, but that was all we allowed. And this included all of the twist knots and all of the knots up to 6 crossings. So this is a broader class of knots than the ones that could be achieved with the twisted hairpin theory. And then furthermore, we looked at the ways that we could achieve these, and we found that there are multiple ways that we can achieve these. We call this model the loop flipping model. And here we listed all the knots that could be achieved. We could also achieve the negatives of these. And we indicated if these are all with RR, and we indicated if it was a plus and the number of twists in A and B. This is the number of ways we could get these knots with loop flipping. So I don't know. Let me just ask when I should stop. Vitali.
Erika, do you have many more slides?
I can go on, but I can also stop here.
Okay. Yeah. Yes. How about some summary or conclusion?
So let's see. So let me give a summary. So the summary is there are lots of interesting types of entanglement that can occur in proteins, especially if we consider disulfide bonds as well as metal ions, both with knots and links and spatial graphs, including thetas, cysteine knots, cyclotides, lassos, etc. And it's not really known why all of these occur, but they're interesting, and they probably do have some function that includes stability because they provide a lot of stability. And how they occur is quite a mystery, but there are various theories for how the knots can fold. And as more and more knots are being found that are more complex, more theories for how they're folding have to be obtained. And these theories are usually developed as a result of first looking at molecular dynamics simulations for how it seems that they might fold, and then trying to develop a general theory of that. And so we're continuing to look at such structures as more knots are formed. Let me just see, I think the last, or the next slide, I should say, is to mention the fact that the 6-3 knot, which has recently been predicted and I believe has now been found and added to the PDB, can't be formed with the twisted hairpin theory because it's not a twist knot. It could be formed with a loop flipping theory, but then it turned out that the molecular dynamics simulations of how it formed did not agree with the loop flipping theory. Wong, Rodden, and I compared it to the loop flipping theories and found it didn't come that way. And then we developed some new theories, which I think I won't go into now.
Okay, thank you very much, Erika.
Are there questions?
Let me stop the recording.